The overall goal of the following experiment is to identify interaction partners of Grb2 by cDNA expression cloning and observation of changes in green fluorescent protein tagged Grb2. This is achieved by first preparing a cDNA library from bacterial lysates. As a second step, the cDNAs are transfected in human embryonic kidney cells, which will result in expression of the reporter construct along with the genes of interest. Next, the transfected plates are analysed on the Opera microscope in order to visualise changes in Grb2 localization throughout the cells. Results are obtained that show epidermal growth factor receptor-mediated recruitment of GFP-tagged Grb2 to endosomes, based on fluorescence microscopy.
The main advantage of this technique compared to existing methods such as yeast to hybrid is that interaction partners of Grb2 can be identified in a physiological situation that's taking into account the subcellular distribution of the interaction partners.
Well, though this method can be used to identify signal transduction by Grb2, it can also be applied to other systems, such as other proteins of interest.
The cDNA library is provided as single bacterial clones in glycerol stocks in a 96-well format. Using the rearray command in the picker menu of the Hudson Rapid Pick Lite, pick bacterial clones from the source plate and inoculate into 1.5 ml LB-amp in a 96 deep well plate. Seal the deep well plates with a gas